good sound? Okay, perfect. Well, first of all, thank you so much to the department and the university, Anand, for inviting me for this lecture. I'm really honored to be here, and thank you, of course, to the Reddy family for setting this wonderful initiative. I think it's fantastic that people will focus on education and science as a legacy, and I am really honored to be here. As Anand mentioned, let's see if this is working. Oh, needs to be on. No. Oh no, I know what it is. That's what it is.
Okay.
Okay. So tonight, as Anand mentioned, I worked at McMaster University, and I should mention my lab is also situated on the traditional territories of many indigenous people land, different from here. But in a way, it doesn't really matter who I am and what I do, because what I want to tell you about tonight is this species of magnetic bacteria, magnetotactic bacteria. And the story is much bigger than me, and that the research I do. So I will mention a little bit if there is time, and I don't drag on too long on other things about my own research on this bacteria. But I really want to give you a broad introduction on what they are, what we know about them, and how they and how we are starting to try and use them for different applications, in particular in medicine. So I got to start working with these bacteria 10-15 years ago. I ordered them because I thought they would be fantastic for putting in undergrad physics lab for pre-med students, because it was like this physical object. It's magnetic, but it's also a biological object, and so they are really at the cross between two disciplines, and they turn out to be way too complicated for an undergrad lab. And we're still working on them, and there are lots of things we don't understand. But what was evident at the time and didn't disappoint is that they really sit at the crossroad between many different science. So as a physicist, I'm really interested in them. Biologists are interested in them. Engineers are interested in them. And you will see that geologists and astrobiologists also have interest in these bacteria. Chemists, of course, and even oceanographers sometimes. So anyway, so they are fascinating object for that as interdisciplinary kind of creature. All right, so let's get started. So I'm going to take you to the world of very small objects, bacteria. They're very small, but I want to start with something very large, which is the Earth, and more precisely the Earth's magnetic field. So I'm sure you all know that Earth has a magnetic field. It's been there for a very long time, at least 3.5 billion years. We're not completely sure. And this magnetic field has one very important function, is that it protects us from cosmic rays coming from the sun. If this magnetic field wasn't there, there would be no atmosphere. It would be stripped off by cosmic rays, and there probably wouldn't be any life on Earth. Certainly there wouldn't be life as we know it. So it has this protection function, but it's also and of course so the Earth's magnetic field is due to the molten metallic core of the Earth, which is creating this magnet. You can really think of it. It's not like a bar magnet, but it's really creating the same type of field as a fridge magnet would, except it's much stronger. So if we try to I try to be quantitative about it, this is the value of the magnetic moment, which is one way we can quantify how magnetic the Earth is, and it doesn't matter what the value is, of course, but this 10 to the 22 here means that this field is about, if you compare it to the magnetic field created by your fridge magnet, you would have to multiply this field 10 times, 21 times. So multiply your fringe magnet power by 10, then 10 by 10 by 10, 20 times, and that's how powerful the Earth's magnetic field is. So So very, very strong. Now, so it has this protective role, so it's very important for life on Earth, and so probably the emergence of life on Earth, it might be linked with the emergence of this field, but we are aware of this magnetic field more for its navigation property, right? So humans have been using the magnetic field for quite a while for guidance. So as far as we know, it was first used in China, so there are records in China several hundred years BC that scholars were experimenting with magnetic rocks that they found, and they had devised some kind of tool that was rotating and aligning with the Earth's magnetic field. It wasn't clear that they used it for navigation right away. They were using it for divination first, but eventually they also realized that it was useful in terms of navigation, and certainly by the Middle Ages in Europe, somehow this knowledge had percolated to the Western world, and the compasses were in use. So compasses were created by magnetizing iron using this magnetic rock called lodestone, which is the most which is the most common type of magnetic rock that you find around, and so that's usually how people started experimenting with magnetic properties, was by playing around with lodestone. So with lodestone, you can magnetize iron, and then you can use iron to make the needle of a compass, and you can find north, and you can find your way around, at least on the north-south axis. So I'm going to keep it very light on the physics, but just so we're all on the same playing field in terms of, how magnetic field and magnet behave. You don't need to know, actually, and you probably know this already, but so what's important for us to understand what the bacteria are doing is to understand what happens when you put a magnet in a magnetic field. So if you have a magnet, like the one that is in a compass needle, this is a material that has a north pole and a south pole, and if you put, as physicists, we describe this as a material that has a magnetic moment. So you can put an arrow that points from the south pole to the north pole, and the length of the arrow might have to do with how strong the magnetic moment is. And again, so the magnetic moment is going to describe how strongly this magnet is going to align with a magnetic field. And so if you place this magnetic moment, this magnet in a magnetic field, this will create a force or a torque that's going to rotate your magnet and align it with the field. So that's something that happens as a consequence of the law of electromagnetism. And so the stronger your magnetic field and the stronger your magnetic moment, the stronger this propensity of aligning with the field. So this is, of course, exactly what happens when you have a compass. So if this represents the Earth creating a magnetic field, and here, if you place a little compass at different points, it's going to align with the line of the magnetic field that are created by these magnets. Okay, so if I'm here, it will point toward the north magnetic pole. So, right. So we use this as a guide. But what's interesting is we've known for a while that animals also use it as a guide. So there's a lot of very solid evidence that birds, especially migrating birds, but also a lot of insects, and also some reptiles, some mammals, some fish, they have this capacity to use information coming from the magnetic field somehow in order to find their way and to be able to come back to where they were nesting last summer or things like this. What we don't know is how they do it. There's a lot of work that's being done in this field, and there are competing theory as to how it happens. And I will mention one of them towards the end, but The reality is we're not sure yet. And different organisms might use different ways as well of using that field. So it might not be just a simple answer. But we don't have a very good understanding of what's going on here, and there are a number of reasons for that. But one big reason is just that they are complicated organisms, and it's hard to experiment on them meaningfully, and so we don't know. But there is one type of living things that is very simple and that does use the magnetic field for navigation, and that we can use for doing some pretty simple experimentation, and those are the magnetotactic bacteria that I want to tell you about. So, so I would say that the reason why those magnetotactic bacteria are fascinating is because they are the simple type of living species that we know of that well, that can be, because the bacteria is pretty much the simplest type of living organism that you can think of that have magnetic property and use them for navigation. So how do they do it? And what are they? So let me give you an introduction to magnetotactic bacteria. And I just want to say that I've really tried to refrain using any type of acronyms in this talk. Scientists love acronyms. I'm just going to use one from time to time, and that's MTB for magnetotactic bacteria. Okay, so if you see MTB, I'm talking about those bacteria. So they were discovered relatively recently. They were discovered in Italy by a medical doctor called Salvatore Bellini, and so that was late 60s and late 50s, sorry, and he was searching for he was looking at bodies of water around the city of Pavia to check if you could find pathogens there. So he was hunting for pathogens, but instead he saw something probably in addition to finding pathogens, I'm sure he found many. He saw something that piqued his curiosity, and he saw some organism that reacted to the magnetic field. So I kind of like this story because it's a very simple discovery. Anybody almost could have made it. So what he did is he just took water from a pothole in Italy, and then he took a drop of this water and he just put it under a microscope. And these are the type of microscope you might have in your schools or in your laboratory. And then he noticed that they were swimming towards. They seemed to be swimming toward the north, even though he was rotating his microscope. I have no idea why he was doing that, but he noticed that. And then he confirmed that they were reacting to the magnetic field by using a magnet that he put beside his sample. And that's it. It found this bacteria. So what he would have seen under his microscope, that's not the image. He didn't record the images, but what he would have seen would be something like this. So it's a little bit unclear, but this would be the edge. So imagine this is a huge droplet. Well, this is a small droplet of water, but it's under the microscope, but it looks huge. This would be the north, where the north pole of your compass is pointing, and here are all these bacteria that are accumulating at the north edge of the droplet because they keep swimming in the direction of the north, and then they are stopped here by the edge of the droplet. So that's what he saw. So he got really interested. He talked about it at his university. He was working as a researcher in a university. He was encouraged to study them, and he studied them for several years, and he discovered a number of things that are. So he found that they behaved as magnets, basically, in the magnetic field. He found that they needed iron in order to have this magnetic property. So probably, however way they were sensing the magnetic field had to do with iron. And he made some really shrewd remarks on a number of things. And one of the things he noticed is, that he immediately saw that the fact that here you had something reacting to magnetic field, it might be connected to, or it might not, but it should be seen in the light of how other species were also sensing the magnetic field. But then, and those of you who work in a university will appreciate it, so he wrote up his results, as you do as a scientist. But in Italy at the time, I don't know if it's still the case, probably not, you had to have the authorization of your university to publish a paper, to send a paper out. And he didn't get the authorization to send this paper. So he just sent it to a few colleagues who weren't very interested, and he moved on to something else, and his discovery got forgotten. So, so publish your papers, or you won't be remembered. So then the bacteria was then rediscovered almost 20 years later, completely independently by an American researcher at Woods Hole, and so that's Richard Blakemore, and so he, again, similar story, seeing things moving in the direction of the magnetic field line, but so Richard Blakemore had something that Bellini didn't have, which was access to an electron microscope. And so he was able to image the bacteria, this magnetic species under the electron microscope. And so an electron microscope is giving you a much better resolution, so you can see a lot more details in what you're looking at. And so here are two different species that he observed around that time. And well, I let you actually, yeah. I don't know if it's possible to lower the light a little bit, or is that complicated?
I can try. I know the button.
Okay. Let a specialist do it. Oh, maybe it's the projector.
You gotta wait. Valerie, just push F on the wall. It's very simple.
Well, okay, this is what happened.
F. No, F. 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 Wait.
Okay.
Oh, that's a good one.
Okay. Now you can sleep and relax better. Okay. Okay, I'm not going to comment on the picture now, but those of you who know what bacteria look like can decide if those ones look any different. But so he studied them. So so leaving aside for the moment, what's different about them, I want to say that these bacteria are very, very common. So they were only noticed relatively recently, but if you go pretty much in any type of fresh or marine body of water, and you collect water at the right depth, and you go back to your lab and you look under your microscope, you will probably see some of these guys. And so they're really not a rare species. They're very common. You find them, so they love water. So they're also sometimes found in sediments. And you find them in weird places such as the human gut. Like a recent study looked at, that there are lots of bacteria in your gut. Well, a small percentage of them is made up of magnetotactic bacteria, probably because they are just so common that we just drink them and eat them and so on and so forth. They can also be very abundant. So if you look at a certain area of a lake, for example, again, certain depths in particular, they might very well be the dominant species of bacteria that you find in that region. So they're a successful species, successful microorganism. And there are very many different types of them. So you saw a few. That was the original one photographed by Richard Blakemore. Here are a few more. They have all kinds of different shapes. This guy lives in the sea. Those ones are freshwater species. This one's very interesting because although it's a prokaryote, so it's a bacteria, it likes to live in little communities of several cells. So it's kind of like a precursor to multicellular organisms in a way. And whether that has to do with the fact that it's also magnetic or not, that's a question that some people are asking, because then you have some specific interactions, physical interactions between cells that maybe keeps them together. So that's one interesting aside. Okay, so what do all these cells have in common? So so if they're so diverse, what is it that, why do we have a single label for all of them? So there's two things that they have and that other bacteria don't have in combination. The first thing, the most obvious, is they have these little crystals here that are magnetic crystals. And those are called magnetosomes. magnetosomes is actually the name of the crystal plus a little lipid envelope around the crystal, the magnetosome. So that's one thing. And the other thing they have, and that you can't see very well, but a little bit. Oh, sorry. Yes, magnetosome. So these magnetosomes make them magnetic. So they are now like little magnets. So remember, we were talking about the magnetic moment of the Earth. So your fridge magnet has a magnetic moment of about 10 in this weird unit here that's on the slide. The Earth has a magnetic moment about 10 to the 22. The magnetic moment of this bacteria is about 10 to the minus 15. So it's like if you take your fridge magnet, you divide its strength by 10, by 10, by 10, 16 times, and then you get the strength of your bacteria. So it's a very small moment. It's nothing, this is not going to demagnetize your room, your hotel key or something like this. But at the scale of the bacteria, it's very significant. So, and of course, it plays a big role in their behavior. So the second thing they have, which is not very, doesn't appear a lot, but maybe you can still see it, is they have here, this one here has a flagellum. So like this very thin thread, which is like it has like a tail, if you want, that it uses for propulsion. This one actually, it has two, but in this picture, it's not a very good, well, it's hard to catch because they're very fragile, but so you can't see the second one. But this species has two flagella, and they use them for swimming. So what all these species have in common is that one, they are magnetic, but two, they are also motile, so they swim. They don't stay in place. Some bacteria don't move, but these ones absolutely move. Like so this propulsion associated with the magnetism is what makes them. That's where the navigation comes in. Like they are going some places, and they are using the magnetic moment to decide where they are going. And they swim pretty fast for something that small as well. So now if you go back to this image, you see here like the line of magnetosome is very evident here. Here in this species, it's there. There's also a line of magnetosome. And I don't know why on this, because of the projector is so good, but this one has two flagella here and there, and this one has one flagella there, and a lot of hair. This is a hairy type of bacteria. But they're also both motile and magnetic. Okay, so fascinating things about them. So one thing that's fascinating is that they make those crystals, those magnetic crystals, right? They make their own magnets. So how do they make them? We know quite a bit about that. So this would be the membrane of the cell. So it's like the skin of the cell. It has a double skin, and you see what happens is it kind of invaginates here a little piece. It forms a little bag, a little balloon here. And once the balloon is closed, it starts accumulating iron inside of the balloon more and more, and it grows this crystal little by little. So and it does this again and again. So it forms several of these balloons and several of those crystals, and then it aligns them. It's hard to see, but here there's a filament. So there's like a rigid rod, and it attaches all of these crystals along the rod so that they are in a line, and so that all the little magnetic moments of each of these crystals align in one direction to form a larger magnetic moment. Those bacteria have a series of genes in their genome, in one region of their genome, so you can cut that part of the genome, and it contains all the genes it needs for this process to happen. So it's like a little program inside the cell, which has been passed on to many different species. Like all these different species of bacteria, they look different. There are different types of bacteria, but they all use the same genes with some small variations. So this is a capacity that has been invented once and has been passed around from bacteria to bacteria. And so those 20, 30 genes together, they are what make this process here of formation of crystal possible. And so from a biological point of view, understanding what those genes are, what they do, how they work together, which one are essentials, which one are not, is very interesting. And of course, can you take this little program and can you put it in something else than a bacteria is also something that's very exciting. And from looking at all these species that have the same program, we can kind of go back in time and take a guess as to when this capacity first emerged. And it turns out that it must have, so bacteria must have learned to do this a really long time ago. It's so well, it's so well spread in so many species. So scientists who work on bioinformatics think this happened billions of years ago. So almost very close to the origin of life. So it's a very ancient capacity here that we see. The crystal they forms are also amazing. So this is a high-resolution electron microscopy image of one of the crystals. So it's about 20 nanometer in size. And you can see it has little facets because it's just like a diamond. It has specific planes that it likes to its faces to follow. You can see every single atom if you look well in this image. And so the structure of this is very well defined. So those are crystals of magnetite, which is an iron oxide material. So it has iron and oxygen atoms organized in a very special way. And I don't want to say too much about this, but if people want to ask questions, I'm happy to talk about it. So magnetite is it's a very common magnetic material. So this lodestone we were talking about earlier that people use to create a very early compass is in fact a form of magnetite. Because it's so big, you tend to have domains of magnetites with different orientation of the magnetic moment. So lodestone is not as magnetic per volume as pure magnetite is, but it's a form of magnetite. And the bacteria, they are making this what's called single domain magnetite, where all the magnetic moment of the iron atoms are pointing along the same axis, and so they add up in order to create the largest possible magnetic moment of magnetite. Yes, and lodestone is a Minecraft type of block that's magnetic and that you can use to build compasses. I read from the internet because I don't play in Minecraft myself, obviously. I don't have time. Yes, but magnetite is again is very common, but the pure magnetite that these bacteria are making is quite special. So another really cool thing about this bacteria, if you think about it, is that they when they die, they don't completely disappear like most bacteria do. They leave their little crystals behind, just like a dinosaur or a whale would leave bones behind. And so that's called magnetofossils. And you can find a lot of these fossils in rock in particular. And so geologists have been using them, not always necessarily needing to know that they come from a bacteria, but as a magnetic marker in rock. So when the bacteria die, and let's say that they become part, their magnetosome become part of a rock, the direction of the magnetic moment of this little magnetosome is aligned with the magnetic field at that time. And so looking at those rocks and looking when the rock was formed and what the direction of the magnetosome inside the rocks are tells us what the direction of the magnetic field was at the time the rock formed. And so it's studying these little magnetic crystals, not all of them left by bacteria, but quite a few left by bacteria, that we can infer variation in the direction of the magnetic field in the past. So that they have a role in that. We can also tell from the exact composition of this crystal. So it's mainly iron and oxide, but you can have other types of atoms that are, mixed up with this perfect structure. You can kind of say something about the chemical environment that the bacteria lived in. So it's another way that these fossils can be used to kind of go back in the past and looked at environment. So, okay, so small quiz to see if people are following. So here are a picture of two types of magnetite crystals, both about, the scale bar here tells you they're both about the same size. So if you were to guess, they were both collected from rocks. if you had to guess, one of them was produced by bacteria, and one of them was not produced by bacteria. It was produced by normal chemical and geological processes. So if you had to guess, which one do you think were produced by bacteria? Who would vote for A? Yeah, and who would vote for B? Are you? So A is the right answer in this case. So the way you can tell, I mean, of course, those of you who answered B might be right, because this is just, it's not that scientists tells us that, or geologists tells us. Anyway, but the reason why we think these ones have been produced by bacteria is that they have these characteristic edges here. They all more or less have the same size and shape, and so they have this kind of quality factor that the other type of magnetite crystal didn't have. The other ones were more dispersed in terms of sizes, like their shape were all over the place, and so on and so forth. So yes, so the fact that we have the capacity to some extent recognize when we find a small magnetite crystal whether it was made by the bacteria or not is something that caused quite a bit of press about 10 years ago, when analyses were done on a meteorite that was found in Antarctica, but coming from Mars. And when people looked at the composition of this meteorite, they found little magnetite crystal, and this is the image of one of them. And you see that it has all the characteristics that we were talking about, with these edges here. It has the right size, and so on and so forth. And so some of the scientists who examined those crystals proposed that this could be the sign that there had been at some point on Mars some of those magnetic organisms, and that they left those fossils behind for us to find, and that this was the proof that there had been life on Mars at some point. So this is quite a controversial conclusion. There's a lot of counter-argument you can make about the fact that it's a few of those crystals that could have gotten there. Otherwise, they might have been formed by processes we don't know. People were then able in the lab to make something that looked like this a little bit by some processes. So I don't think it's like, I don't believe this is a proof, like a solid proof that there was life on Mars. But just to keep in mind that those are very abundant, and they are in terms of finding traces of very simple organisms. They might be our best chance to prove that such organisms were in a certain place. So it could be that in the future, that looking at magnetite crystal and understanding where they come from is one of the ways that we could say something about whether life was present in a very simple form in different places in the universe. Okay, so another quiz. It's the last one, I promise. If I were to ask you where the magnetic field points in this room, what would you think? Yeah, you can show me with your hand. Yeah, so yeah, I see some answers all over the place. So I don't know. I'm completely lost here, but I brought a compass, and so this compass tells me that north is more or less in this direction. Now, this is a trick question, because the north is in this direction, but the magnetic field actually points down. It points north for sure, but it's not something we think about because we don't need to think about it, but it also points down. And this is important for us because that's what matters for this bacteria. So north, but also you can see it on this image, especially so it points down if you're in the northern hemisphere. It's going to point up if you're in the southern hemisphere, and it is going to point to be horizontal if you are at the equator or close to the equator. So I looked it up, and so in St. John's, the inclination is 66 degrees. So it means you're 66 degrees below the horizontal for the magnetic field. So that's certainly not, it's not negligible, so down. So why does it matter? So the question we haven't addressed or answered yet is why would this bacteria want to align with the magnetic field? Like for us, okay, we use north as a way of understanding where we are. For the bacteria, so those bacteria, another thing they have in common is that they have very, very specific oxygen requirement. So if they grow in a place where there is too much oxygen, they feel very sick and they die. If they grow in a place where there is not enough oxygen, they also don't fare so well, and suddenly they're not able to make this magnet, which is partially made of oxygen. And so where they want to be is they want to be in this very special zone. So if this is a lake, and this is the bottom of the lake, and this is the top of the lake, and if you look at the oxygen level, so there is a lot of oxygen at the surface because it's being dissolved from the atmosphere. But as you go down, there's less and less and less oxygen. And there is this very special Goldilocks zone here called it's another acronym, but it's the oxic-anoxic transition zone. And that's basically where the oxygen disappears. And they want to be right there because they have a little bit of oxygen, but not too much. And so if you're a bacteria, and you're pretty much, so you think you're a bacteria, you're pretty much this small bag of water. So, and you're floating in a lake. So your weight is completely negligible compared to other forces around you. And anyway, you're made of water, so you're buoyant. So you don't feel what's up and what's down. Some bacteria have developed ways to detect light. So they will know where is up because that's where the light is coming from. But those bacteria usually don't have this capacity. But they use the magnetic field to be able to go down or to go up, depending on, so they're able to sense oxygen as they go. And so if the oxygen level is going in the right direction, they'll continue swimming in that direction. If the oxygen level is right, they might stop swimming. If it's going wrong in the other direction, they might reverse direction. So a lot of bacteria, a lot of organisms go about, I mean, we all have places that we prefer, and we prefer to be in some places than others. And a lot of organisms, maybe surprisingly, go about finding those places in a random fashion. They just walk around until they find the right place. But here, being able to swim up and down allows them to be way more efficient in finding this region, which probably means that they have a really strong requirement to be exactly in this region here. And so that we think that it's why they have this, developed this capacity, and it seems to be such a precious capacity that it's very widespread around many different species. So that's why they are magnetic. So I'm just going to do a little parenthesis. Not that I have a lot of time, but so it's my second time in Newfoundland, and the first time I was there, I was really looking forward to visit L'Anse aux Méduses and see the site where Vikings had landed. And I drove there, and I had a wonderful guide. Like the young man who did the visit for our group was absolutely fantastic. And by the end of the tour, he mentioned that he was actually a graduate student, and he was working on bacteria that were processing iron. And I got really excited, and I thought, my God, he works on the same bacteria as me. So it turns out that no, there are other types of bacteria that process iron. So there are bacteria that precipitate ions, and they tend to be very common in places like L'Anse aux Méduses that have bog iron in the water. And so those other bacteria play a major role in the ion cycle in this environment. And of course, we know that the Vikings liked this place in particular because they were in the business of smithing iron. And so it's no coincidence that they're in a place where there were rivers with iron and with bacteria that process iron. Now, I don't know that anyone has ever tried to find magnetotactic bacteria in Newfoundland, or if they did, they haven't published any paper that I've read. But I suspect that if anyone was going to go there and fish at the right depths in the water, you would find a lot of magnetotactic bacteria. So, you know, if anyone feels inspired to do so, please contact me, and I would love to facilitate this process. Okay, end of the interlude. Back to fully magnetotactic bacteria. So I think I've taken a lot of time for my first part. So my second part was going to be a little bit more about my own research. So I'm going to skip about it quickly. You can ask me about it if you're interested. I'm interested in how they swim. And so doesn't necessarily have to do with their magnetic property, but the fact that they are magnetic allows us to have a lot of control over them so that we can make measurements on them that are very precise. And so what we like to do, okay, so let me just show you all the movies I had in that part. So this movie is a classic. And this, what you see here is a sample of blood. So the round cells are red blood cells. The big guy that's crawling around is a white blood cell, so he's trying to defend the bloods against pathogen. And the little black things that look like alters are bacteria. And so the white blood cells is chasing after the bacteria, and it's got it at the end, and it's eating it. And so this is a classic movie to show cellular motions. And the point I want to make here is that cellular motions at the scale of bacteria and cells look very different from the motion at our scales, because the type of forces that cells are exposed to on the small scales are very different from what we experience on the large scales. And so there are, okay, so this is magnetic bacteria in the magnetic field. So they are going really fast in this movie. It's coming back, and one of my students has tracked them, so you can see their trajectory. And here, what I wanted to show you is that they don't swim perfectly along the magnetic field. There is some noise, right? The trajectories are not perfect. And so one of the things that's, equations. But I'll go over them very quickly. One of the things that's different at small scale is that noise is important. There are thermal motions. Things are very agitated. And so let me just skip all the quantitative stuff and say that if you're at high temperature, or if you have a weak magnet, then the bacteria are kind of aligned but not perfectly. And if you have at a very low temperature or a very strong magnet, then they would be perfectly aligned. And you see that in the case of our bacteria, we're in this case here. They're aligned, but it's not perfect. So that's one thing to take into account. The other thing to take into account, the other thing to take into account is something called Reynolds number. So Reynolds number is, and let me again skip all the details, but Reynolds number is a quantity that you can calculate for any situation of something moving around a fluid. And what enters this equation is the velocity of the thing that's moving, its size, its density, and the viscosity of the fluid. And depending on the value of this quantity, it's going to tell you if, at the scale of your problem, inertia dominates or viscosity dominates. And so if you have a problem where viscosity dominates, then the type of flow you will have is called laminar, so that volumes of fluids that are side by side tend to have the same type of velocity and direction. Whereas at high Reynolds number, this tends to be a turbulent flow, and weird things happen in your fluid. Now, just to give you, without again skipping all the calculation, if you calculate this number for a penguin, you find a very big number. If you calculate this number for a bacteria, you find a very small number. And so this is just to illustrate that what we experience as, sorry, this is a puffins. I've changed it because I'm in Newfoundland. What we experience as objects the size of a puffin is, if we swim and go to the swimming pool and you swim, you're going to create a turbulent flow as you swim. And so in order to swim efficiently in a turbulent flow, you better be streamlined to avoid too much turbulence, and this kind of put constraint on how you should be swimming. But the constraints are very different for bacteria because they have a very small Reynolds number. So for bacteria, it feels like swimming in honey, basically, or even worse than that, in something very, very, very viscous. And so the strategy you would employ as a bacteria to swim are very different from the strategy you would employ as a larger animal. Okay, so the good strategy for swimming in viscous fluid is the corkscrew strategy, where you rotate a helix, and as you rotate the helix, you're being propelled forward. So it works for a corkscrew. I'm sure you can all imagine what happens when you use a corkscrew. It works for pasta in a fluid. You can see that the rotation is quite pronounced. As it's being translated down by gravity in this case, it's forced to rotate. And this is exactly what happened for our bacteria. So this is not our bacteria, but it's bacteria that were imaged. It's another type of bacteria, very known. It's not magnetic, but you can see its flagella very well, and you can see this beautiful helical shape that's being rotated as the bacteria moves forward. So that's how a lot of those bacteria move forward. Okay, let me skip all of this. And this is our own species of bacteria. It has two flagella, one at the front, which we don't really know yet what it's doing, but one at the back, which is being used as a corkscrew. And in our case, the body is actually also like a corkscrew. So it's like you have two corkscrews attached together with different handedness. And there's a little motor right here that rotates one against the other. And that propels everything forward. And this is how they work. People have, you can make some of these, like with a little rotational motor, and you put them in water, and you can make little artificial bacteria this way, and it works well. Okay, so I just want to show a picture of my students because they do all the work. This is Shariful, this is Lucas, and they had a really hard time growing these bacteria because they are so sensitive to oxygen level and iron level. You have to be it's actually a lot of work to work with them well, which is why they're not appropriate for undergraduate labs, by the way, if you're wondering. And this is Paolo, and you can see here at the microscope, and these are the electromagnets we're using to generate a magnetic field. So he's working at the moment on trapping those bacteria in a little pits so that we can observe them for a very long time. So here is trapped bacteria in liposomes. And here is trapped one single bacteria in a little pothole, and could you look at it for a really long time. Okay, so that's what we're playing around in the lab. And so let me skip this old part and just go and tell you about what we can do with them. So very quickly, I want to conclude. Right, so they're magnetic. This means if you change the magnetic field, you can change their direction. So we do this very often in our lab by switching. So here the magnetic field constantly reverses. So when the bacteria arrive at the end of the sample, we just reverse the field on them, and they have to make a U-turn. And so this way we keep them in the field. But so what this means is that in some way they work like a remote control car. And you know, if you have a setup that is a little bit more sophisticated, you'll be able to decide where they are going, right? So the idea is that, okay, these bacteria or similar ones, you could use them as remote control little nanobots. So this seems like a crazy idea, but it's actually not. So here is a movie from a group in Montreal that was made about 10 years ago. And so the idea here, let me play this. Okay, so they're working with this type of magnetotactic bacteria. And instead of using a single bacteria, they use a swarm of bacteria. So here they have a magnetic field that concentrates the bacteria in a single spot. And then they use this swarm for moving things around. And so here, this here is like a huge flock of magnetotactic bacteria. And they are using them to move this little brick in order to construct a pyramid. So in the interest of time, I'm not going to play the whole movie. But little by little, they build this very simple pyramid. And this got a lot of attention in the field as the demonstration that cells can build pyramids. And you can make them do it. Now the pyramid itself is a bit underwhelming when you see it. It's only a one-dimensional or two-dimensional pyramid. It's very small. It has only a few bricks, but it was still like a really cool idea. So this one, 10 years ago, Now this group in Montreal, they have a spin-off company, and they've designed a proprietary type of magnetic cells. And this is from a movie on their website, like you can go and watch the whole thing. So they've designed these cells by modifying their genomes, probably their magnetotactic bacteria initially. They have somehow attached some little vesicles on their surface that carries drug. And what they do, they're at the moment doing clinical trial to send those magnetic cells, swarms of them, just like they did with the pyramid. They inject them at the location of tumor, and then they are using very strong magnetic field, a little bit like in an MRI machine, to concentrate the cell exactly around the tumor, send them at the center of the tumor, and then release the drug. So this is advertised as the way of doing better chemotherapy, where instead of having drug all over your body and destroying cells you don't want to, you're really able to concentrate the drugs exactly where you want to. So this is, I think, an application that sounded really crazy 10 years ago, but is really now taking form and right here in Canada, and might change the way we do some type of cancer therapy. So I think that's one cool example. But there are many other things there are many crazy ideas you can think of with this magnetic property. So I mentioned you could take those genes, make something else magnetic. So people have started doing that and using cells that are easier to manipulate than the magnetotactic bacteria, cells like E. coli. So you can make them magnetic, and then you can direct them. In this case, this group was using them to design sensitive tests, because they can send the bacteria with their detecting capacities in the right position on their plate. But another thing you can do if you make cells magnetic is you can use them as contrast agent in MRI experiments. So MRI detects magnetic moments is one of the things MRI detects. And so if some of the cells in an organism were to produce magnetic particles, they would really pop up in your image. And so, so a lot of groups are also working toward doing some genetic modification to cell lines in order to be able to improve magnetic resonance imaging. So again, we have collaborators at Western University in Ontario, and they are trying to do exactly that. So it's something I think will also happen in the coming years. And you can also another idea is just to use those particles in isolation, because I mentioned that they were very high quality. And so instead of using the whole bacteria, which is clunky and hard to grow and something like this, you can just grow a lot of bacteria, kill them, harvest their magnetic particles, and then use the particle for something you want to do. And one thing you can do, for example, is you can, by exposing those particles to very high frequency magnetic fields, so quickly varying magnetic field, you can make them heat up very strongly. So you can create very local changes in temperature with those particles, and that's another way you could help destroy tumor if you're able to target them in the right place. So that's possible application. There's a lot of other things you could do with this small particle. You could embed them into soft materials, for example, and created little soft objects that you can remotely control, and that's another area of strong research at the moment. And I think we're going to see a lot more of these small magnetic robots in the future. And so keep your eyes open. Some of them will have to do with magnetotactic bacteria, some of them not, but certainly some of them will have to. Okay, and I want to leave you on one last slide, and one last idea. And it's something that was discovered a couple of years ago by a French group, and I think is really cool and really exciting. So they were fishing for magnetotactic bacteria, as one does, in the Mediterranean Sea. And what they found is a magnetic organism that was not a bacteria, but was a eukaryotic cell, so a more advanced type of cell. And when they looked at it closely, what they found is that this cell, in fact, it's a cell called a protist, so it's a motile type of eukaryotic cell. And it was magnetic because on its surface, it had magnetotactic bacteria attached to it. And so what they discovered is an organism that lives in symbiosis with magnetotactic bacteria. So the organism itself doesn't have magnetic property, but it has hijacked the magnetic property of the bacteria. And when it divides, it separates the bacteria into two. So now they really live as a symbiotic organism. And so I think this is the first time this was observed, and this gives us an idea that maybe one possibility for higher organisms to sense magnetic field is that there's some kind of symbiosis with simpler organisms that do produce magnetic crystal. So again, something that's not proven at all, but that has been proposed as one possibility. Okay, and with this, going back to the birds and the bees and the magnetic field, I'm going to thank you for your attention. Thank you so much for coming here and listening about this organism. And yes, and if you have any question, I'm very happy to answer them. Oh, and if you want to play with magnetite, I have some crystal of magnetite here at the front.
So I believe this lecture is being recorded, so please wait to get the mic before you ask a question.
Who got questions?
They're going to run around asking the mic. Questions?
Bet.
I don't need the mic though.
Please use it because then it gets recorded. Thank you.
So that bacteria is amazing, and I can see in the lab we can control it with applied magnetic field, but in the environment, in the nature, the only magnetic field is the Earth magnetic field. So are they continuously swimming to the North Pole? Do they end up there? Thank you.
They don't end up there. No, because I think just like all of them mostly are in bonded bodies of water, right? Like maybe they swim to north of Lake Ontario. But first of all, it's not that they always swim north. It's they follow the line, but they keep switching back and forth between going north and going south because they are just, they're just following until they find the right place. But then if they overshoot, they'll go the other way. So they don't, and also I think they are so small that in their lifetime they wouldn't move very far, and I guess in water there would be other type of forces like waves and currents that would completely overwhelm the small efforts that they make going in one direction at a time. But yes, but it's a good question. Like, yeah. I think also it's possible that they are not magnetic all the time. So in the lab, for example, if you grow them and you don't give them the right food, they'll stop making those magnets. And then if you start feeding them well again, they'll start growing them. So it's possible that in nature, they are magnetic for a while, then maybe they are not, and they are brought back somewhere by currents, and then they start again. So it's not they are constantly swimming north, I think, yeah.
Hi, great talk. So you're talking about you can find these things at, these certain depths in puddles and that. Because of what you were just saying, does that mean that they aren't as likely to be found around the equator because that wouldn't help them find the right depth, right?
Yeah, and that's what people were. There was a hypothesis, like in the beginning, is that they wouldn't be found at the equator, but they are found at the equator, and so maybe they just ended up there because they were on their way north, or they was being brought by currents, or it's possible that it's possible that they are not using this for navigation. So some people have alternative explanation for why it would be, let's say, or maybe in addition to navigating, maybe it's used for other things. So one other thing that's been proposed is that they use this as a kind of battery. So it can generate chemical energy by oxidizing the surface. So I'm not exactly sure about the details here, but it's been proposed that this could be used as a reserve of something. I mean, we know that other species are accumulating ions for other goal as well. So it could be that there are alternative roles, and so even if you're at the equator, there's maybe some advantage for making those magnets. But yeah, that's an interesting question.
Okay, and I have another question if nobody else has got one.
Yeah, go ahead.
You were mentioning about the meteor, that meteorite, and the idea that it may say something about Mars, but certainly Mars does not have a magnetic field now, and I don't think it's had a magnetic field for a very long time, which is